Hi, everyone. Welcome to today's Journal Club. I'd like to thank ESGO and IGCS for their support in our Journal Club. Before we begin, to mention a few housekeeping notes relevant to the Zoom platform for today's Journal Club. We're recording today's discussion, and the recording will be made publicly available. Participants with cameras turned on may appear in the video recording. So we ask that everyone please keep your microphones muted so that we can hear the questions in the presentation. And if you're having any difficulties or questions, please send a message in the chat and our technical support team will assist. We definitely will have time for questions and answers and encourage you to submit your questions via the chat platform throughout the session. So with that, then I want to get on to this month's lead article titled Genomic Alterations, Molecularly Targeted Therapy and Survival, a real-world endometrial cancer molecularly targeted therapy consortium cohort study by my dear friend and colleague, Alvarez Secord, and her team. And she's at Duke School of Medicine, Department of Obstetrics and Gynecology in the Division of Gynecologic Oncology in Durham, North Carolina, here in the United States. So I'm very, very pleased to welcome Angeles back to our podcast and journal clubs. Angeles, thank you so much for joining us today. We're really looking forward to this discussion. And please proceed with your presentation.
Thank you so much, Pedro, for that kind introduction. And I'm so excited to present this journal club on behalf of all my colleagues that helped with this important research endeavor. And thanks all of you for being here today. So I'd like to just move to the next slide and just talk a little bit about why we had this objective and really why we were prompted to do this research. So as you're aware, endometrial cancer continues to rise in incidence and mortality around the world. In fact, there was over 400,000 cases reported in 2022, and almost 100,000 individuals died of this disease. And we know that those numbers are even higher now today than they were. The US data just came out for anticipated cases this next year. It's about 70,000 cases and 13,000 deaths. So I think it's really important that we understand this perspective of this disease from the global impact and what we can do to help individuals all over the world. So one of our goals was to confirm the proportion of at least one actionable genomic alteration. And why we wanted to do that is because the modalities that we have, radiation and chemotherapy, were really antiquated at the time that this study started. And even though we were combining them both together, we weren't really helping women live longer. And so we felt that if we could identify that there were targets present and that if therapeutic treatment directed at those targets improved survival, that would be really meaningful. So our other really important part of this study was to determine if targeted therapy is associated with improved survival. So in terms of methods and how to do this, we all love to do research. Many of us do our single institutional studies, but we wanted to do something on a larger scale. And so we had an opportunity to bring together sites through a grant we received to develop a multidisciplinary consortium, which we call the Endometrial Cancer Molecularly Targeted Therapy Consortium. Good job, Pedro, for saying it at the beginning.
I was very proud, actually.
Yeah, it is a tongue tripper. And we call that EMCT2 or EMCT squared to make it easy. And it was really formed to study tumor testing and treatment for individuals with advanced and recurrent endometrial cancer. So we had 9 founding sites. We've now grown to 23 sites. And what I'm presenting to you here today is, the initial aspects of our study. And we had about 12 sites enrolled in this trial. And we sought to abstract data, including age, stage, grade, histology, race, ethnicity, genomic alterations, protein expression for HER2, P53, mismatch repair, hormone receptors, and survival outcomes. And then we conducted a statistical analysis. We had a statistician that was involved in the development of the study and does all the work on the study, which has really been helpful. And our patient demographics, our median age was 64 years. And I want to just point out importantly, in terms of the racial distribution, 24% of our patients identified as Black, 68.5% were white, 2% were Asian, most were non-Hispanic. So we're very excited about this because a lot of the clinical trials include a much smaller percentage of individuals who identify as Black. and they're much less likely to represent a diverse patient population. So we have a lot of improvement to still make here, but we felt like our representation was a little bit higher for some of the more diverse groups. And 67.8% had recurrent or persistent disease, and then the remainder had advanced disease at the time of initial diagnosis. And we identified, so we wanted to do this as clean as we could. This also happened during COVID. So our initial plan when we developed this consortium, that a monitor would go out to each of these sites and review all of this data to make sure that the data source was correct, which is a huge undertaking for an observational study. However, with COVID, we weren't able to do that. So we had to kind of figure out another way to ensure that this data was as clean as possible. So we had everybody submit their tumor testing, their pathology to us for a second review to make sure that the data was accurate. And so we had over 1,100 patients initially in the study. You included 967 patients that we felt that the data was as clean as we could possibly make it, given the constraints that we had at the time. And of those, 902 or 93% had tumor testing of some kind. 576 had NGS testing and had at least one genomic alteration in 11 pre-specified genes. We can come back to that. There's always a lot of question of like, why did you limit it? But, again, it goes back to the fact that this was a grant. We couldn't be on a fishing expedition. We had to limit what we felt were the most important genes. And the most frequent genomic alterations were PI3K at 36%, TP53 at 35%, and PTEN at 26%. Now, of those patients, 233 received 292 matched biologic therapies with a median follow-up of 29.7 months. And for these patients, what we did is had a separate individual at those sites review the data in duplicate to ensure that they were correctly entered. And then any dispute was adjudicated either at our Duke site or at their site. And for the match therapy group, the progression-free survival was 6.9 months. And the median overall survival was 20.5 months. And when you compare that to what has been shown in the literature, which cytotoxic chemotherapy, the medians are typically 3 to 4 months, and the overall survival times are like around the 12-month range. Next slide, please. So in conclusion, we felt that the consortium facilitated the development of real-world data on genomic testing and molecular targeted therapy. And also it allowed us to evaluate patients from more diverse populations, which we feel is really incredibly important across all of our cancers. And then survival appears to be improved for individuals receiving matched biologic therapies compared to historical controls. Next slide. I just want to say and share that this was a huge, huge team effort. We received the grant from the Kay Yao and Jimmy V Foundation for cancers that affect women. And these are two huge basketball heroes in North Carolina. And then we also have a picture of my hero at Duke, who's Coach K. All of these coaches exemplify a strong team spirit, and through the team spirit here, through our initial founding sites, as well as our expansion sites, you can see all the people that played such a role in getting this work done. And I want to give a shout out to our Duke research team, Bonnie Thiel, Nikita Bish, Carson Smitherman, Lauren March, Kristen Weaver, Jesse Paradis, and Sarah Lynn Hurt. Without their assistance, this never would have been even attainable. To all the patients who participated in the study, and to all the researchers and their teams at all these various institutions. And thanks, Pedro, for letting me share this data with all of you today.
Angela, thank you so much for that presentation. And of course, I just actually want to say really congratulations to you. You have been such a leader in the field of gynecologic oncology, and having you, certainly with all of our fellows in the journal, you're such a role model. And I'm so happy and proud that we kind of like both of us moved along at the same path. I should say that Angeles and I met for the first time during my first GYN oncology interview for fellowship. So we were in the waiting room, and that's when we met. So I'm really, again, so proud to see how you have really shined throughout your career.
Oh, that's very kind of you. I certainly don't feel like a role model, so I feel like it's just everyone just joining together, being part of the team to really help our patients. That's what drives us, and I'm really thrilled to see as the next generation comes forward how we can help all of you achieve your research goals, and also just help these individuals with these cancers and preventing cancers.
Well, thank you so much. And again, I see that there's over 20 participants already and in the platform. So I wanted to first start with a few questions myself. You know, you mentioned what a unique opportunity this was. 12 sites, a huge consortium. Can you just give our listeners and our fellows your perspective of like how do you get all those people together and how do you actually achieve a grant to do this?
Yeah, so this idea actually started in 2017. So 2017 was when we started hearing about the data from PORTEC-3 and GOG-258, which I think we were all very excited and thought, hey, if we do chemo and radiation together, we're going to cure more patients, you know. And then the results from that study didn't necessarily show that across the whole group. And so it became clear that we need to start thinking outside the box. Just because we have chemo, just because we have radiation, those aren't necessarily the tools that we should be using. And then also at the time, we have the TCGA data coming out saying that endometrial cancer is much more complex. It's now these 4 subgroups. I actually think it's even far more complex than that. I bet that these 4 groups are going to be split into further groups. And then we start having some of these trials reading out where we have a biomarker agnostic approval for immunotherapy. We have the trastuzumab studies released by Dr. Fader and Dr. Santine. We have some translational research data that Dr. Doug Levine presented from 86P, and we have Dr. Slomovitz's work with everolimus and letrozole. And then we have Dr. Fiorico's work with migestrol and tamoxifen, and the ability to do NGS testing in the clinic. And so at 2017, when you think back, like, you know, now when we present this data, it's like, yeah, not a big deal, of course. But 2017, this was kind of a big idea. But we couldn't get to work, we couldn't work with drug companies to blend this all together into one big study, you know? And also because some of these drugs go off patent, they're not as interested. So we had a lot of obstacles. And so part of getting it done was networking. I happened to sit by Dr. Bhavana Pathuri, who we've also known for a long time, right, Pedro? And we were both frustrated with the lack of progress in endometrial cancer. And so we both had similar ideas. And so that kind of started it. And then it was just reaching out to friends really from these large institutions to see if they'd be willing to partner with us. And then just one day got this email from the Duke Cancer Institute about this grant. And I was talking to my mentor and boss, Dr. Andrew Burchak, who had started an ovarian cancer consortium called OCAC, which is an international consortium that does a lot of great work. And so I was asking him, like, I can't do this. You know, I said, there's no way we're going to get this funded as a clinical trial. Can I do something like what you did with the consortium? And so he gave me the roadmap with the ovarian cancer consortium to make this happen too. But you can imagine like getting the grant approved was a step. Then you finally get it approved and then boom, COVID hits. So I guess if I have to impart anything, it's just when you find a barrier, just keep finding a way to overcome that obstacle. Just it's about solutions, it's about resilience. Don't ever give up, you know. And so I think that's what the biggest picture about. And then when we started to get successful in presenting our data, other people wanted to come on board. And Pedro, you know, I'm not a no person. I'm a, you know, like, yes, let's explore, let's find ways to work together. So anybody who wants to come join us, we don't have any money at this point in time. Our grant is actually, I have to submit my final report June of this year. But we're always happy to work with others.
Excellent. So thank you so much. And you heard it here. If you want to go visit Duke, it's a beautiful area of this country. So North Carolina is wonderful. So as we typically do, we have our fellows asking you questions. And again, we also encourage those who have joined to ask questions through our chat. So for the fellows, if you can just state your name and where you're from, and then please provide your question. So, Luisa.
Yes, thank you, Pedro. So good evening to everyone. My name is Luisa Popper. I'm from the University Hospital Münster in Germany. And thank you for this interesting presentation and insights to your work. So during the course of the study since 2017, the standard of care for endometrial cancer changed significantly, right? So what is, in your opinion, the main finding that we can learn from of the study, despite the absence of this standard of care during the course of the study?
Well, so I think one of the things, I'm going to go back from standard care for a second. I think one of the things that we find really challenging with our clinical trials is making sure that we engage and enroll individuals from diverse patient populations. It has gotten better. The most recent trials have about 10% of the patients identify as Black, compared to before that was 5% or less. But by doing a real world consortium study, you're able to maybe engage more diverse populations in the research. And also whatever you identify in the clinical trial may not be generalizable to the real world population. So efficacy may not be quite the same. Side effects may be worse in a real world population. So by establishing a consortium, you're actually able to take some of those data elements from a clinical trial and then evaluate them within the context of this consortium to see, is efficacy the similar? Are there any new or different safety signals? So that's one thing by just establishing the consortium. The other thing in terms of the standard of care changing, yes, you're absolutely right. Like, talk about like a huge swing from not as much interest in targeted therapy to, can you imagine a world without targeted therapy in endometrial cancer right now? So my impression is absolutely individuals who have endometrial cancer need a biomarker assessment. How big that should be right now, I don't have the answer for that, because not every alteration is targetable, or not every alteration is actionable. Let's just say it that way. It may be something we can target, but we don't know if it works. And I think the other aspect here that we haven't figured out is, how do we sequence patients on therapy? Like, I struggle with somebody who's has HER2 3+ disease in the frontline setting. Should that individual receive a trastuzumab-based regimen or immunotherapy? We don't have the answer for that yet. And I think sequencing is a huge question that we need to address.
Excellent. Thank you so much, Angeles. Angeles, you mentioned about, I think I heard you said several times, obviously, the impact with regards to diversity and how you were able to capture so many patients of diverse backgrounds. Can you tell us a little bit as to how did you strategize to achieve that?
Well, we're still trying to achieve that, because I think we've done well in increasing representation for individuals who identify as Black, but not so much for patients who are Hispanic or individuals who identify as Asian. So part of it is reaching out to sites that have different patient populations. So during this interim time, Hawaii came on board. So we're hoping that that will help obtain more information for patients who identify as Asian or Pacific Islander. And then we also have other sites that have a higher percentage of individuals who identify as Hispanic or Native Americans, another huge unmet need. And then at our center, I think it's very important to make things as easy as possible. So we have consents that are translated for individuals who speak Spanish, and also we can do this over the phone rather than people having to come into the clinic.
Excellent. So our next question will be from Frederica.
Hi, everyone. My name is Frederica. I'm a fellow from the Heidelberg University Hospital in Germany as well. Thanks a lot for your presentation. My question is, given that several therapies like PD-1 inhibitors are now approved regardless of the MMR status, do you think that the 6-month rate of 54.5% in the biomarker matched group might overestimate the value of molecular testing?
Oh, I don't. I guess that's the easiest response. No, so when we looked at our methodology at the time, I think we declared that we would have to see 35% to reach what we felt was statistically significant. And so 54%, I think, was very strong. And also what we did in terms of the advent of immunotherapy, because that was a big change in the standard of care, we actually evaluated the groups in 2 separate cohorts. We did a sensitivity analysis for patients who didn't receive immunotherapy and those that did. And we found some similar outcomes in terms of PFS rate at 6 months and median PFS. Where things changed is in the landmark analysis, where that seemed to benefit more patients with immunotherapy. But the bottom line is, I think that there's a value to knowing what these biomarkers are beyond mismatch repair status and beyond TMB and MSI, because those other biomarkers are not just targetable, they're actionable.
Excellent. So we're actually going to jump over to some of the questions on the chat. And this one's from Albert Jacob. Thank you, Albert, for joining. Congratulations on a great study. Question is, were the genomic alterations in the 11 genes the same or was there significant variation? Did the kind of genetic variation affect survival? Also, what was the incidence of more than one gene mutation?
So this is actually an area that we have driven into only in terms of P53 and not the other ones. So excellent question there. And I think we do need to go back and evaluate how many patients had more than one genomic alteration, because I don't think we've done that yet. For P53, what I can tell you is there's a variety of different alterations that are present. And in terms of the different alterations that we're seeing, we did not notice a difference in survival. But now as you start to slice down into those numbers, we're talking about limited numbers. You're also talking about multiplicity and data analysis, and we did not do adjustments for this data as it was. So everything would have to be taken with a grain of salt, because I would not be very confident in the validity. However, I do think what you're talking about is the wave of the future. It's not just about if there's a mutation or not, but the biological significance of that mutation and what the function is.
Excellent. So we'll come back to other questions on the chat, and we'll next head over to Mariana.
Thank you, Dr. Pedro. Congratulations on your presentation. And my name is Mariana Gouveia. I'm from Brazil, Hospital 9 de Julho at São Paulo. And how do you interpret your results, considering that retrospectively included patients, most of whom had recurrent disease and had potentially received multiple lines of chemotherapy and underwent this molecular testing significantly later in the disease course compared to a prospectively included patients? Do you think this may have confounded the study results or had any impact?
Oh, yes. A type of study like this could definitely have confounding results because there's going to be bias, right? So the type of testing, the type of treatments, that was all determined by the investigator. There could also be bias in terms of who they put on the study, right? So maybe they're thinking, oh, this patient's done really well. They're thinking about the study, but a patient who may not have done as well, that patient didn't get put on the study. So, again, I go back to what this really means in terms of where we are today in treating patients with endometrial cancer. And I would just say the data points to the importance of knowing the underlying biology of the disease, identifying and testing for biomarkers. I think by testing for biomarkers too, we can actually hopefully have greater equity in terms of the treatments that are offered to our patients.
Excellent. So next, actually, we'll go back to Luisa, and she's going to ask the next question.
Yes, thank you. So in the study, we saw that more than a third of the tumors had P53 mutations, but only little of these had targeted treatment for this alteration. So did these patients receive treatment based on other targetable mutations of their tumors, or are there any therapies which might be useful in this specific group of patients?
Right. So when we added P53 in, the interest there was WEE1 inhibitors. And also there was some translational data from 86P. It was a 3-arm study that evaluated different combinations with either chemotherapy alone or with bevacizumab or ixat-vip alone. And temsirolimus was the other drug involved there. And from the translational research, it appeared that patients who had P53 alterations may have some benefit from the addition of Bev. So there's kind of a twofold reason to include P53. There was just a small number of individuals who got a WEE1 inhibitor because it's not clinically available. It's only in a clinical trial. And most patients didn't get P53 directed therapy. I mean, we'll see what happens in the future. There's been a tremendous amount of interest in development of drugs that target P53. I think one of the issues, though, has been toxicity. There is a trial coming forward, though, that's going to be evaluating the use of bevacizumab in a prospective manner in patients who have P53 mutations. I can't remember the study design of that trial, and I don't know if it's in the public domain yet, but it is coming.
Great. So Angeles, let's go back to the chat, and this one's from Arthur Shu. He wants to go back to the example that you gave of the patient. He says, to follow up on your statement with regards to the patient on HER2 positive endometrial cancer, do you do chemo and trastuzumab? Do Do you do chemo and immunotherapy, or do you do a combination of 4 drugs?
Oh my gosh, Arthur, this is like in every debate right now. It's like I'm going to throw it back to the whole crew and see what you would do. Okay, so we have GY026 open, and I'm strongly recommending to all my partners, and I hold myself accountable to that too, that all those patients should be offered GY026, which is a 3-arm trial evaluating trastuzumab containing regimens in 2 of the arms. The phase 2 study that was done by Dr. Nichols, Fader, and Santine, we were a site at Duke, so I was very lucky to participate in that study. But I think it's important to know that even if a phase 2 study is randomized, it still is a design that can be flawed and there can be bias. And it's incredibly important to confirm those results in a rigorous phase 3 trial design. So my answer is a clinical trial every step of the way. Now, without a clinical trial, this is a huge conundrum, right? Because what is the best treatment for these patients? And then also, one of the things we found in our sensitivity analysis is that the patients who received immunotherapy ultimately had the better outcomes compared to the non-immunotherapy group. But again, it's just you just have to be so cautious how you interpret that data. So this is an area I'm just very thankful that we have a clinical trial, but I definitely struggle.
So, Natalia?
Hello, I'm Natalia Teixeira. I'm working at Hospital Clinic in Barcelona. And I was wondering, did you evaluate the cost effectiveness of the next generation sequencing and tumor testing in your data?
No, we did not evaluate cost effectiveness. The issue, these tests are super expensive, right? So NGS tests range anywhere from $5,000 to $6,000. If you do the smaller IHC testing platforms at Duke anyway, it's a few $100s for each of those. If you do pole sequencing with single gene pole sequencing, that's around $600. So absolutely, we need to take into consideration the cost of the NGS testing. And how can is it better to go broad and get all this information, or is it better to have like a super endo, like you would call it an endo chip, where you have the most important and targetable alterations that we know about right now, and you get that data? And I just don't know the answer to that. However, I do believe that doing a cost-effective analysis within the context of a trial is incredibly important.
Absolutely. Absolutely. So we'll turn now to Sofia Bigardi.
Thank you. I'm Sofia Bigardi from the University Hospital of Padova in Italy. And my question is, did you record whether any patient received 2 different immunotherapies after progression on the first one?
We do have some information about those patients, but it's actually a pretty small number. I think I have to go back. There might be in that one slide. There were, I think about 250 some that received match therapies. 233, we have at least 1 match therapy. So it was just a really small number that received more than one. So we did not go in further and do like a subset analysis of that group.
Okay.
Thank you.
So, Angela, going back to the topic of race, and that question in the chat is, given that this consortium shows more racial information, is P53 mutation more prevalent in a certain race? And what is your experience in P53 IHC immunohistochemistry staining versus NGS?
So we've actually reported on that both in this paper and outside this paper. So, and others have reported this too about P53 alterations, even going back to, my mentor, Dr. Bertrek, did some of the initial reports of P53 probably in the 1990s, when they were doing IHC testing, right? So it is individuals who identify as Black, individuals who have serious cancers are more likely to have P53 alterations. And so that wasn't something new. In a follow-up study, we were able to show that survival outcomes were similar in those groups if, patients who have P53 alterations did bad no matter what. Now, other groups have shown that even within the patients who have P53 alterations, Black patients still tend to do worse. So I think some of that's just going to be what you find as academic centers. Is that generalizable to all centers? And then in terms of staining versus NGS, a lot has been reported here as well, with some groups showing high concordance rates. However, when you look at those studies that were done where there's high concordance rates, there's usually a specialized gynecologic pathologist, and a specialized gynecologic pathologist is not available everywhere. So in a follow-up paper that we did and Carly Whalen published, I think our concordance rate was in the like 0.7 range, which is still good, but it's not 100% concordant. So my feeling is if you have P53 overexpression, that is really strong. That is easy to say you have a P53 mutation. If there's any situations where it's equivocal, or you have, because absence of staining doesn't necessarily, may portend a null mutation, but in those situations, it may be worthwhile to do NGS testing just to make sure that you do or do not have a mutation. So there's, at our site, there's like 4 different expression levels for P53 that our pathologists evaluate. Overexpression, decreased expression or absence of, and then there's the normal wild type expression.
Very interesting. So we'll turn over now to Marina Rosano.
Thank you. My name is Marina Rosanu. I'm a fellow at the European Institute of Oncology in Milan, Italy. You stated that 69.4% of the patients received paclitaxel and carboplatin as a frontline chemotherapy. Did you record which type of therapy the remaining 30% of patients received?
So these patients were comprised of a group with advanced and recurrent disease. In those situations, the patients with recurrent disease may have just received radiation or even no additional therapy. So it wasn't like all of these patients were candidates for systemic chemotherapy at the initial diagnosis. And to answer your question directly, no, we did not record that.
Excellent, very well. And now we'll turn over to Australia. Helena Obermeyer, she'll ask the next 2 questions, I believe.
Hello, my name's Helena. I'm a gynae oncology fellow at Westmead Hospital in Sydney, Australia. Dr. Alvarez Secord, did you find a difference in patterns of targetable mutations found in endometrioid and non-endometrioid tumors?
Well, we did not do that in this study. However, we did another study that was recently reported at the Winter SGO meeting by Dr. Glenn Boyles and Dr. Victoria Bay-Jump, and they did find a different pattern expression there, and they also evaluated it in the context of race as well. So there does appear to be differences. I can just tell you from that study, endometrioid tumors are much more likely to be characterized by PI3K, PTEN alterations, much more likely to have higher estrogen receptor expression, and deficient MMR, MSI high tumors. The serous cancers are much more likely to have P53 alterations. And the other thing that they recently reported in our patients from the consortium was that Black patients were much more likely to have alterations. I think it's in FBX, I can't remember the whole acronym, and BCL. I think it was BCL2. So yeah, there's a lot of differences there between the histologic subtypes and a race. And don't quote me on these other things because now I'm going beyond this study.
Oh, don't worry, it's being recorded. So
Just as a disclaimer, we have gotten off the topic here.
And Helena, you had a second question?
Yes. The newly revised NCCN guidelines for uterine cancer suggest consideration of germline or multi-gene panel testing for all patients diagnosed with uterine cancer. If you added germline testing for patients in your study, would you expect to see more therapeutic targets?
Interesting. So germline testing, we actually do reflex germline testing based on deficient MMR. I would have to look into that data further. I mean, I think part of this goes back to what Natalia asked about in terms of cost effectiveness. And so I think we need to take that into consideration. And so I'm a little hesitant to recommend germline testing for all. I mean, like I said, we basically do that on the MMR and MSI data. I'd like to look at the guidelines further and see why they're making those recommendations. And then in terms of the genomic testing, okay, so this is my research. I'm a huge fan. I'm a biomarker nerd. I love biomarkers. NGS there's newer technologies coming down the pike. I'm hoping that over time NGS can become less expensive. And then I think that the newer opportunities that we have to delve into, like proteogenomics, are going to change the landscape even further. At this time right now, for individuals who have advanced or recurrent disease, I think NGS testing is very important, but I also want to be cautious because I think there's expense there. And as I mentioned before, if we could get down to an EndoChip right now where we could say, this is what's the most important thing that we need to dive it down to, because these are the drugs that we have to target this, I would be in favor of that. But again, this is, when we're talking about here, this is policy. These are guidelines to direct therapy for everybody, and the implications that has on the cost and so forth is pretty huge. So I feel a little hesitant to advocate strongly for that. I think we need to be precise in our recommendations. So I'll tell you what I do: P53, MMR, HER2, ER on everyone, and then POL. I tend to use a more selective approach with POL. Our data from the consortium showed that 1% of our patients had a POL alteration. It's just not that high compared to what's been reported in other studies. And this is an advanced and recurrent group. So I've tend to move more toward a selective POL testing where I feel like it's going to make a difference in how I treat those patients, or can they go on GY032? And I think PORTEC 4A, the results just being released from that are really interesting, and that may expand a little bit further what we decide to add in terms of our testing, but I like to see the paper first. So I tend to be, I just tend to like to be more directed in that approach rather than just getting everything. But having said that, I think there's a lot of value here, but we also have to take a look at the cost.
Brilliant. So, Angeles, we'll just take one additional question from the chat, and of course, leave it to Arthur to ask the really simple questions. Arthur, again, who has every GOG study memorized, he says, data from GOG 86P states that bevacizumab performs well in P53 mutated patients. When do you use this regimen in the current paradigm of treatment?
So, Arthur, I do not. I love the study was a great translational study. I think it gave us a lot of information, but I'm not ready to utilize it in clinical practice yet. I think it's so important that we study it. And this is one of the reasons we wanted to do the consortium, to study this question. And I'm hopeful that the trial that I talked about earlier will move into activation, and I will enroll patients without hesitation.
Excellent. And there's one last question. Certainly, obviously, as you see, we have a global representation. And my question is, how do you see the results of this study being applicable in low and middle income countries? How do we take this information and hopefully bring some benefit to those patients? Uh-oh, I think we lost the Wi-Fi connection. Arthur?
Oh wow. Yeah, we after
We'll have to check with Ashley. All right. Well, I hope she will be coming on back soon again. But in the meantime, while she's coming back on, I really want to thank the fellows who participated. You all did a fantastic job. Thank you so much for putting your questions together. I hope you enjoyed this experience as part of the fellowship. Angeles is actually texting me now. She says, I didn't like that question, so I just disconnect. No, I'm kidding. No, she's telling me that she's trying to connect back in.
Sorry. Sorry about that. Yeah, it was like it just abruptly stopped and I could not reconnect. It's the craziest thing. It was like Pedro must not like my answer, so he disconnected me.
No, I was actually saying that you had just texted me and then you said I didn't like your question, so I just disconnected.
Not true at all. I don't know what part I fell out, if you heard me at all, but
no, we didn't hear you at all. So yeah.
So what I was saying, I actually don't use that regimen in clinical practice. I think the 86P study was an incredibly important study. It's an ancillary study, and it helps us inform future trial design. And as I mentioned, there's a trial that's plans on evaluating this very question, and I will enroll patients on that study without hesitation.
Yeah, Angela, the question that I was closing with was for our international audience, particularly low and middle income countries, how should they take this information?
Oh, well, this is where this is one of the things I love about your journal club, Piers. You get to meet people from all over the world, and this is a global problem. And this is where I think we all need to work together to ensure that there's equity in terms of the care that patients have access to and the care they receive. I don't have a great answer for this, but I think it goes back to what is the primary therapy that patients should be getting up front now. I probably have some different thoughts about this, like in terms of advanced patients who have measurable disease. Yes, immunotherapy, I agree with that. Patients that don't have measurable disease, who don't have a deficient MMR, MSI high tumor, I'm not going to do immunotherapy. So how can we boil this down so that there's a certain set of biomarkers that are integral into the practice of care and ensuring that everybody gets that testing done?
Great. Well, Angela, once again, thank you so much for taking the time out of your busy schedule to join us to have this discussion. Thank you for your loyalty to our journal and really sending very important work to our journal. And congratulations to you and everyone who participated in that consortium. And once again, my hat off to you for all that you have contributed to gynecologic oncology and what you continue to contribute. So thank you.
You're so sweet, Pierre. Thank you. And thanks all of you. Wonderful questions.
Excellent. So I do want to thank again all of the fellows. Thank everyone that joined for the Journal Club. And I want to remind everyone to join us in June for our Journal Club. It'll be with Jason Wright, somebody Angeles and I know very well. And he's going to be talking about trends in modified radical hysterectomy in patients with less than 2 centimeter cervical cancer in the United States. Of course, obviously, very timely as we have the results of the SHAPE trial recently. So we hope that you will join us for that. Have a great weekend, everyone. And thank you once again.
Bye.